Welcome to the Witnessing Lectures. In this video, we are going to talk about the pathogenesis of HIV. So HIV stands for Human Immunodeficiency Virus. Okay, so HIV is the causative organism for AIDS. Okay, and then this virus belong to a family of RNA retrovirus. They are referred as retrovirus because these are type of virus that reverse transcribe their RNA to DNA once they enter the cell. Then their DNA also will transcribe into a messenger RNA which will make proteins and the new virus are made. Okay. Then apart from that, HIV has different types. So we have type 1. Then we also have type 2. Type 1 is more prevalent as compared to type 2. So this is a structure of the HIV virus. Okay. So this HIV virus, it's enclosed in a lipid bilayer membrane. Okay, so this is a lipid bilayer membrane, as you can see. Then this lipid bilayer membrane, it's embedded with the glycoproteins. Okay, so we have this glycoproteins, which is here. This is a transmembrane. So this transmembrane glycoproteins, it's called the GP41. Okay, this glycoprotein, it helps in the fusion of the membrane of the virus into the membrane of the CD4 cell, which we will discuss in the replication. Then on top of this same GP41, there is a docking protein which is called the glycoprotein GP120. Okay, so this GP120, this is the protein that assists in the attachment of this HIV virus to the target cells. Okay, so this other diagram is showing this is the CD4 cell because the HIV virus targets the CD4 positive cells. So we have different types of CD4 positive cells. We have the T helper, we have the macrophages, we have the monocytes, the microglia, as well as the dendritic cells. So this GP120, it's the one that interacts with the CD4 positive cells. Now, this same GP120, it has got some clinical importance. The clinical importance that is there is that why is it that despite so many studies, scientists have failed to come up with the vaccine for HIV? It's because this GP120, it undergoes several genetic mutations, okay, which has made scientists to fail to come up with the vaccine against the HIV. Apart from that, the GP120 and the transmembrane, they are produced by the gene which is called the envy. It's called the envy gene, okay? So it's called the ANV gene. So this envy gene, it's the one that produces the GP120 and the GP41. Now, as you enter this virus, there's another protein which is called the matrix protein. Okay, we are not going to talk so much about this matrix protein, the P17, because it has no much clinical importance. Okay, and then again, next to this matrix P17, as we enter inside, there's a capsid. So inside the capsid, that's where we find the genetic materials for the virus. Now, this capsid, it has 20 sides, okay? So it has got 20 sides. That's why it is called an icosahedral, okay? So it has got 20 sides. Then this same capsid, when the virus enters the cell, the antibodies that are produced by the host cell, they are against this same capsid, which is made up of the proteins called the P24. So it is the antibodies against the P24 which are detected when you do the HIV test, such as the ELISA or the Western blot. Okay. Then the protein, the matrix P17, and the P24 proteins, these are produced by the gene which is called the GAG gene. Okay. Now, as you enter inside this capsid, you find two strands of identical copies of RNA which has the positive polarity. Okay. Then apart from that, you also find the three enzymes. Okay. We have reverse transcriptase. Then you also have the integrase enzyme. Apart from the integrase, we also have the protease. Okay, so these are the enzymes that are found inside the capsid, as well as the two identical copies of RNA. The three enzymes are produced by the gene which is called the polygene. Okay, sorry, this is called a polygene. Okay, then now let's try to look at the replication of the HIV virus. Okay, so we have the HIV virus, which is here. Remember, then we said that this HIV, it has got the glycoproteins. It is embedded with glycoproteins. This one on top is the GP120, and we also have the transmembrane, which is GP41. Okay, so once attaches occur, the GP120 is going to attach to the CD4 receptor of the CD4 cells. Okay, so once it attaches, it undergoes some conformational change. Okay, so this conformational change, it's going to express some domain-like hands. Okay, so these domain-like hands, they are the ones that will interact with the core receptors. These are the core receptors, the CXCR4 and the CCR5. So these will interact with this harm domain, allowing the cell, the membrane of the cell and the membrane of the virus to be put close, okay, so that they can come close. So now we have different types of core receptors. For example, the CXCR4 core receptors may be present in the T helper cells. Then the CCR5, mostly they are present in the macrophages. But the macrophages, they are mostly infected as compared to the T helper cells. So once this virus, the membrane, is put closer, then the transmembrane, which is GP41, it will undergo some change. Okay, so this GP41, it's going to slide. So it's going to slide so that it gets it gets closer to it's going to slide so that it's the membrane between of the virus and the membrane of the CD4 cells will fuse. Okay, so this is what this other diagram is showing. Okay, so if we have if this is the virus, then there's interaction between the glycoproteins and the co-receptor molecules. Then this will allow entry of the virus content into the cell, because the membrane of the virus and the membrane of the cell, they're fused. So once the content of the virus enters into the cell, the capsid is going to dissolve. Remember, we talked about the capsid, okay? So once this capsid dissolves, the capsid, it's going to release the 3 enzymes, which is the reverse transcriptase, then it's going to release the integrase as well as the protease. Okay. Then apart from that, it's also going to release the 2 strands of RNA. Okay. So reverse transcriptase, it's going to start the RNA. Okay. So once it starts the viral RNA, then it's going to transcribe. Okay, there will be reverse transcription of the viral RNA to viral DNA. Okay, so this is what happens. So if, let's say, this is the viral RNA. So this viral RNA, it's going to transcribe, okay, into the viral DNA. Then reverse transcriptase, it's going to cut, okay. So it will cut, then so that the DNA will remain alone, okay. So once this complementary DNA it's made, then again it will undergo a replication, in which it will become a double RNA, okay. So once this double RNA is made, the double-stranded RNA viral DNA, sorry, it will leave the cytoplasm. Okay, this is the cytoplasm. So it's going to leave the cell cytoplasm and goes into the nucleus. Okay, there in the nucleus we have the enzyme integrase. So the enzyme integrase, it's going to cut the viral DNA. Okay, so it's going to cut it. Once it cuts it, then it's going to integrate it into the DNA of the host genome. Okay, so once it is integrated, then the DNA becomes a pro-viral DNA. So it will become a pro-viral DNA and it becomes part of the host DNA. So this is a very crucial step in the production of the new viruses. That's why once the viral DNA, it is integrated into the cell of the host genome, then the person becomes HIV positive for life. Okay, now we have the viral genome. Okay, so this viral genome, what will happen is this. So this viral genome, okay, this is the viral genome. Okay, so this viral genome, it has got the ends here, okay, because it has been integrated into the host for the host genome. Okay, so this side we have the host genome, this other side also we have the host genome. Now this viral genome, it will express itself at the end here. It has got what are called the promotory region. Okay, so it's from this promotory region. These promotory region, they are the ones that enhances transcription of this viral DNA to messenger RNA. So once it is transcribed, then it will express itself. The viral DNA, it will express itself. So it's going to have, it has got 3 genes, okay? So the first gene, we have the GAG gene, okay? This GAG gene produces the P24, the P27, and the P17 proteins, okay? These are the proteins which are produced by the GAG gene. Then we also have this other gene, which is the polygene. So this polygene is the one that produces the 3 enzymes: reverse transcriptase, protease, and integrase, okay? Then also we have the envy gene, okay? So this other gene, it's the one that produces the glycoprotein, the GP120 and the GP41, okay? Now these proteins, the way they are produced, they are produced just as a single strand, okay? So they are produced just as a single strand, okay? Okay, so once they are produced, these proteins, we have the enzyme, which is called the protease. The protease, it's going to cut them. The example that I'll give, for example, from the GAG gene, where the P24 and P27 are produced. So these proteins will be cut, okay, because they are unfunctional. So it is after when they are cut, that's when there will be different proteins. So this enzyme which does this, it's the protease, okay? So you have the protease. This is the enzyme which will cut these proteins. So these proteins now, they are going to assemble. So once they assemble, then the new virion will be formed, and an immature virus will be formed, which will bud out of the cell and get matured. So this, once it's mature, then it is going to infect other positive CD4 cells, okay? So there will be billions and billions replications of the HIV virus. Thank you so much for watching this video. Don't forget to subscribe to our YouTube channel, or as well as to follow or like our page, so that you'll be able to receive a lot of updates.